So today we are talking about cellular respiration. So there's lots that could be said about cellular respiration. We're going to actually take two full lectures to go through all the details on this. And what I will try to do is point out which details are the most essential, because there is a lot of information that we're going to cover in the next two lectures. So first of all, let's just go back to remind ourselves of what we were talking about before the break. We were talking about energy and ATP and this thing that we're calling cellular work. So cellular work, there's some examples here. You can see that basically we're taking ATP, we're breaking it down to ADP and inorganic phosphate, and somewhere in the middle there's cellular work going on. So what does that look like? There were three examples that I gave. One is moving things across membranes. So this is usually pretty obvious to people when you're moving things. This is cellular work because you're actually moving things, and moving things sounds a lot like work. Another example is using motor proteins. So in this example, you can see we have a motor protein and it's moving a vesicle along a microtubule. This could be chromosomes along a microtubule. This could be motor proteins in your muscles. That's mechanical work. The biggest type of cellular work falls under the category of what we call energy coupling. So maybe I'll write that down here, energy coupling. And what does that mean? It means you have a reaction that is endergonic. So it requires energy to proceed. It's not going to proceed without that input of energy. And so we can input ATP. You can see they're showing that in the diagram, how a phosphate is actually donated to the X molecule. And that gives it energy, it promotes it, it puts it in a higher energy state, and so now it can react with Y. And that is what we need ATP for. And our muscle cells, believe it or not, consume something like on the order of 10 million ATP molecules a second. Either that or it's a minute. Either way, it's still pretty impressive. So we need lots of ATP, and that's where respiration actually comes in. So let's talk about respiration. The whole idea behind respiration is that we want to take ADP right here. We're going to combine it with an inorganic phosphate, and we're going to make ATP. But this takes energy to do so. So how do we do this? We want to harvest energy from our food and our nutrients, and we're going to do this in a number of steps. And this is what we're going to talk about. So there's two ways actually to make ATP in our body. And we're going to kind of just talk about this just a little bit. If you take further biochemistry classes, it's important you know a lot more about these two processes. But there's two ways to do it. One is called substrate level phosphorylation. The other is called oxidative phosphorylation. So let's take a look at these two things. So substrate level phosphorylation, what does that mean? It means you have a substrate. So what is a substrate? A substrate is a molecule that's being acted on by an enzyme. And so this substrate is going to take a phosphate the enzyme is going to take a phosphate from the substrate and it's going to attach it to an ADP. You can see that's what's happening here. Basically, it's removing a phosphate from here, attaching it to ADP, and we're making ATP. And that's substrate level phosphorylation. So why is it different from oxidative phosphorylation? I'll get into that in a second, but this is one way where we can make a little bit of ATP during our respiration processes. So oxidative phosphorylation is a little bit more complex, and this is what is happening in the inner membrane of the mitochondria. So this is going to involve a proton gradient. It's going to involve a specialized enzyme called ATP synthase, and you're going to have a gradient across that membrane. And this whole process is called chemiosmosis. And we're going to talk about this a little bit later. I'm not sure if we're going to get there today. If not, it'll be we'll talk about it next day. But we probably won't get to the mitochondria, at least this part of the mitochondria today. So a couple other definitions to throw at you in terms of some chemistry. When we're breaking compounds down to harvest the energy, often these are involving what are called redox reactions. So oxidation and reduction reactions. So if you take a look at this, I'm looking at this little thing right here, oxidation. So oxidation is where a compound loses electrons. So you can see that this compound here, A, has a couple of electrons, and then it becomes electronless over there. The opposite reaction is reduction. So B here is gaining those two electrons and becoming reduced. So So that's the terminology. Hopefully everyone knows that terminology. I think a lot of people have seen this little memory method. So Leo the lion says grr. Leo means loss of electrons is oxidation. Gain of electrons is reduction. So it's a little, whatever it's called, a numogram to help you remember what you're talking about. So let me show you some examples of some oxidation that it makes a little bit more sense when we're actually looking at chemical reactions. So it's very easy to understand oxidation and reduction reactions when we're looking at the formation of ions. So if you take a look at this, we have sodium. It's got it's a neutral charge, and we've got chlorine, which is a neutral charge. And so you remove an electron from sodium, it becomes Na+. So this becomes oxidized. The chlorine gains the electron and it becomes reduced. So in that case, it's usually pretty easy to understand this because you can actually follow the electrons. But in terms of what is going on with organic compounds, unfortunately, it's a little bit more complex. So let's take a look at a very simple organic compound. We'll look at methane here. So here's methane, and methane is made out of carbon and 4 hydrogens. And one thing you may know about carbon and hydrogen is when they form a bond together, the bond is nonpolar. So what does nonpolar mean? Nonpolar means that they equally share the electrons. So maybe I'll write that down, nonpolar. Equally share electrons. So in that case, the carbon and the hydrogen are equally sharing their electrons, and that's what we have here. Now, if you take a look at this reaction, we are oxidizing methane with oxygen. Okay, so there's some molecular oxygen right here. And our final compound is carbon dioxide. Now notice carbon dioxide, it has oxygen and carbon, and this forms a polar bond. So remember, the polar bond is an unequal sharing of electrons, unequal electron sharing. So in this case, if you take a look at where they're showing where the electrons are, oxygen is sharing them more. It means it's using the electrons more than the carbon is sharing. Oxygen is electronegative atom. And so what's going on here? It means carbon doesn't have the electrons as much. So carbon is losing the electrons, and so carbon is becoming oxidized. Oxygen, on the other hand, is gaining more electrons, and it's becoming reduced. And so remember, electrons are part of chemical bonds. They're very seldom are they loose and doing their own thing by themselves, but they're part of chemical bonds. And so in this case, we're oxidizing methane. So we're going to use that terminology to say we're oxidizing fuels that we eat. So we eat glucose, for example, and we oxidize it. And this is what is happening in terms of the carbon is not seeing those electrons as often. So I know it's a little confusing because you can't see the electrons, they don't seem to be going anywhere necessarily. But if you take a look at this and understand that the polarities are changing, then the amount of sharing is changing quite a bit as well. So let's take a look at glucose here. There's our glucose. It's being oxidized. And so a similar reaction is being combined with oxygen. And we're making carbon dioxide. So the oxygen becomes oxidized and the oxygen becomes reduced and ultimately forms water in the end from that reaction. So one more kind of definition that I want to throw at you, and it's going to be important for understanding respiration, is understanding that these oxidation and reduction reactions are often done by certain types of molecules. And these certain type of molecules, we can call them electron carriers. Electron carriers. And what these electron carriers do is they take those electrons from the bonds. And those electrons are attached to hydrogens. So the term we use is dehydrogenation, meaning we're taking hydrogen away from the carbon. So take a look at this example here. We have an organic molecule, and we've got a hydrogen here and a hydrogen here. And we have this thing here, this electron carrier called NAD+. I'll show you what that looks like in a moment. And this NAD+ is going to take one of the hydrogens. So NAD+ or NADH, well, I'll talk about NAD+ in a moment, but you can kind of think of it as like this. So this line is a bond, and that bond is 2 electrons, and H represents hydrogen. So in this case here, what we have is the hydrogen plus its electrons are taken. The other hydrogen just becomes a free ion, and we can kind of ignore that. It's not doing anything at the moment. But notice that our carbon has become oxidized. It has less electrons, or at least less electrons on the carbon, but it's also losing those hydrogens. So usually in biological reactions, when we're looking at oxidation and reduction reactions, we're looking at dehydrogenations going on as well. So you might see that kind of type of terminology as well, particularly if you take any biochemistry classes. So let's take a look at this NAD plus for a second, okay? So NAD plus looks like this, and the full name you can see is nicotinamide adenine dinucleotide. So if you look at this compound, this here is actually adenosine, the same adenosine you'd see on ATP or ADP. And in fact, if I put these together, it really is ADP, but it's got an extra carbohydrate here, and it's got this little thing here, and that is actually derived from niacin, which is one of the B vitamins. And so it's a big long word, and I just want you to remember NAD plus, okay? And so what happens is that NAD plus, you eat your food, and there's an apple, for example, and the NAD plus is going to take those hydrogens plus the electrons from some carbon bonds, and it's going to form NADH. So NADH is shown over here. If you take a look, there's the hydrogen. So just a little bit of chemistry for you there. So the overall reaction looks something like this. The NAD plus takes the 2H plus 2 electrons, it becomes NADH. I guess that's not the overall reaction, that's more or less a half reaction. The other half of the reaction is involving the oxidation of the food, of course. So what I want you to think of this thing here, this NAD plus is an electron carrier. And what it does is it carries electrons in chemical bonds. Like I said, just remember that electrons are rarely loose on their own. Those electrons are almost always in chemical bonds. So NAD plus is one type of electron carrier. There are some other ones, and I think I have some of them listed on the next slide. Here they are. So NAD plus is kind of one of the main ones. We're also going to see another one called FAD, and I think I have a slide showing you how that works here in a moment. And when we get to photosynthesis, we're going to see a third one called NADP plus. And notice in each case, these pick up a hydrogen. Or in the case of FAD, it picks up 2 hydrogens, right there and there. And they're just involved in different processes, and these are all electron carriers. Okay, so, oh, there's FAD. I knew I had it here somewhere. So that's what FAD looks like. And notice that FAD is also basically, there's ADP, and then it has this funny group on there, which is flavin, which is another derived from another B vitamin. And if you watch where those nitrogens are, the ones that are in blue, we end up with a hydrogen here and the electrons, and hydrogen here plus the electrons. And so that's what it's doing. It's carrying electrons. And those electrons ultimately have energy in them. And that's what we care about. And that's why we have these things, because we care about energy. Okay. There's overall reaction for FAD. And it's taking those electrons and storing them in chemical bonds. Okay, so back to this, right? Making ATP. This is really what we care about, making ATP. And so what I want to do is get into and start talking about the processes of making ATP. So here's a breakdown of the steps of cellular respiration. And what I'm going to do is make some notes as well on the whiteboard and kind of go through these processes. But let's take a look at this. We're going to talk about 4 processes in total. So the first one is glycolysis. So glycolysis is right there. It's breaking down glucose to pyruvate. Second step is called pyruvate oxidation. And this is breaking down pyruvate to acetyl-CoA. The third step is called the citric acid cycle. You may have heard of it called the Krebs cycle. That's an older term for this cycle, and you can use either. And the citric acids, or citric acid, I went to the dentist recently. My tongue has just been tied since then. But the citric acid cycle is going to take the acetyl-CoA, and it's not shown here, but it's basically converting it to carbon dioxide. And notice we're making a little bit of ATP along the way. and we're making a whole bunch of these electron carriers. So we'll talk about those soon. And then, of course, we have our last step, which is oxidative phosphorylation, which includes the electron transport chain, and we're going to make a whole bunch of ATP there. So what I want to do is break down these processes and talk about them in a bit of detail here. So the first one is glycolysis, but before I go to glycolysis, I want to make some notes here. And so let me just switch to the whiteboard here. Okay, so there's the whiteboard. And so what I want to do is we're just going to talk about glycolysis for starters. And but before we get there, there's three things I want you to pay attention to. Three things to know. Okay, one, I want you to pay attention to the carbon. Okay, so we'll call number one is carbon. Okay, and I'm going to color code all of this. So I'm going to use a red for number 2. Red is for ATP. And then I'll use blue for the last one. The blue is for the electron carriers. Okay, so those three things I want you to pay attention to as we go through respiration. And what I'll do is I'll probably make part of this diagram today, and the other part I'll make on Wednesday on the actual whiteboard. So let's just take a look at glycolysis for a second first, and then we'll get into the details of glycolysis. So glycolysis starts off with glucose. So let's write down glucose here. So glucose, oops. There we go, glucose. And glucose is 6 carbons. Okay, and so what we're going to do is we're going to break down glucose and we're going to make 2 pyruvate. Slide this over a little bit. And pyruvate is a 3-carbon compound. All right, and so this process here is called glycolysis. Now, there's a few other things to know about glycolysis. Like I said, we want to follow all these different molecules. So number 2 is, of course, ATP. So in glycolysis, we're going to make 2 ATP. So I'm going to just draw an arrow like this. We're going to make 2 ATP. Now, in order to make ATP, you need the ingredients of ATP. So that means you need 2 ADP plus 2 inorganic phosphate. All right, and the other part is these electron carriers. So for glycolysis, we're going to make 2 NADHs as well. So I'll put an arrow down here. 2 NADHs. And to make NADH, you need 2 NAD pluses. And that is my very rough summary of glycolysis. Now, one thing to notice about glycolysis is the name is glycolysis. Okay, so glyco means carbohydrate and lysis means breaking. And we're literally breaking glucose in half. We're going from a 3-carbon compound, from a 6-carbon compound to 2 3-carbon compounds. So what I'm going to do is I'm just going to come back to this in a bit, but that's the first stage of respiration that we want to talk about, and we're going to look at that in a little bit more detail right now. Okay, so there we go. Back to this. So there's the first part, glycolysis. Okay, so let's take a look at glycolysis. So here's an image from the textbook, and you can see they're breaking glycolysis down into 2 components. They're calling the first half the energy investment phase, and the second the energy payoff phase. So you're probably looking at this and thinking, wait a second, I thought there were only 2 ATP involved. Well, if you take a look at the overall reaction of glycolysis, we are going to have to invest 2 ATP, and then we're going to make 4 ATP. So if you think about the net amount of ATP produced, we are making 2 overall, right? It costs us 2, but then we get 4, so 4 minus 2 is 2. And that's really where the balance comes from. You may also notice there's water included, but I don't care about water. Those 3 things I care about, carbon, ATP, and electron carriers. Water's not one of them. So don't worry about water. I'm not too concerned about balanced equations in Biology 107. If you want to balance equations, that's awesome. It's just not important for Biology 107. And you can see the net down there is basically everything I talked about. We've got the carbon, we've got the ATP, we've got the NAD pluses and NADHs. So we'll take a look at these and we'll take a look at the specific reactions. So let's take a look at the reactions of glycolysis, because I want to talk about those in a little bit of detail. And I want to just talk about some of the things that are going on. Now, you do not need to know all the chemical structures. You do not need to know all the enzymes for this. There is one enzyme that I'm going to want you to know for this pathway, but we're looking at 10 reactions basically is what we're looking at, and they're quite complex in a sense. But if you do take biochemistry, you will need to know all the reactions. But for this class, you're looking at what's going on with the carbon, what's going on with the ATP, and what's going on with the electron carriers. So here's our first step, glucose. So glucose is a great molecule. We need it. It's the only fuel that your brain can consume, by the way. So we have to have glucose in our diet, although most of us probably eat too much of it, but it's essential for our diet. And so this is why we look at glucose. It's kind of one of the basic fuels. So this very first step in glycolysis, we're going to take some ATP, and there it is. And so what are we doing here? Why are we adding ATP to the system? Aren't we trying to make ATP? Well, this is basically an endergonic reaction. In order to get this process going, we need a little bit of activation energy. So that's what this ATP is doing. So this is our activation energy. And so that's kind of important. So you go from glucose to something called glucose 6-phosphate. You're adding a phosphate group. You can see the name of the enzyme, hexokinase. After this step, it kind of mutates and changes. You're changing the rearrangement of the atoms, but not much is changing other than that. And what you're doing is you're making this from glucose into fructose 6-phosphate. And so fructose 6-phosphate is a little bit more symmetrical and easier to basically break into two. And that's why we're doing this. So we're adding a little bit more activation energy at this stage here. And this enzyme here, phosphofructokinase. So phosphofructokinase. We are going to be talking about that one a little bit later today. I have some notes I want to make about phosphofructokinase. It's the one enzyme that you should know. And at this point, we've not done a lot other than consuming a couple of ATP. But that's why this is called the energy investment phase, okay? So let's move on and talk about what's going to happen now after this, because after this, we're going to actually get a bunch of energy out of these molecules. So there is one more step that's kind of in between the two stages, and this is the actual breakdown step. You can see that it's happening right here, and we're making two compounds. We're making one called glyceraldehyde 3-phosphate, and we're making dihydroxyacetone phosphate. So by the way, this one here, G3P, or glyceraldehyde 3-phosphate, this here is actually the product of photosynthesis as well. Product of photosynthesis. So we will come back and talk about G3P a little bit later on. I guess at the end of the week, we'll talk about it. Okay, so let's break this down. We've got these three carbon compounds, and at this stage, what you'll notice that this dihydroxyacetone phosphate can easily be converted to G3P. And notice that everything after this has a number 2 in front of it, because it's trying to balance the equation. So I don't care so much about the 2s, but that's why those 2s are there. You can see they're all over the place, these 2s. So it means that all these reactions happen twice per every glucose molecule. So the next step, you notice it gets oxidized using our electron carrier, NAD+. So NAD+ gets reduced, it gains 2 electrons plus a hydrogen and becomes NADH right there. And our resultant compound there, you can see it's bringing on another phosphate there. But if you take a look carefully where the reduction has happened, it's actually happened with some water molecules. But I'm not going to go through all the chemical details on this. We'll move along here. By the way, this here is going to go to the electron transport chain layer, which is something we're going to talk about next lecture. At this point, we're going to actually start making ATP. So here is an ADP going in. It's grabbing this phosphate right there, and we're making some ATP. So this here is ATP being made by substrate level phosphorylation. Substrate level phosphorylation. And we get something called 3-phosphoglycerate. So moving on, this 3-phosphoglycerate it mutates, so atoms get rearranged. We lose a water molecule, nothing we really care about for Biology 107. But the last step, we go from something called PEP, phosphoenol pyruvate, and that last phosphate gets deattached, and we're making 2 more ATP. And in the end, we get pyruvate. So there's our compound pyruvate. And there is the energy payoff phase overall. So these are the products: 4 ATP, 2 NADH, 2 pyruvate. We're also making some water, but we don't care about water. So back to this slide here. This is really some of the most essential information to know, is basically the overall reaction of glycolysis, which I drew for you on the whiteboard, okay? Like I said, you're consuming 2 and you're producing 4. 4 minus 2 is 2. So overall, that's kind of the important part. So before we get to this, I want to go and talk a little bit about that enzyme I was telling you about. So just give me a second here. I got to find my notes. What did I do with my notes? There we go. Or sorry, I got to find the correct whiteboard. That's what I'm looking for. And where is it? Oh, here we go. Okay. Sorry, my computer mouse is just doing something weird for me here, but I think I can do that now. There we go. Okay, we want to talk about that enzyme, the phosphofructokinase. So let's just talk about glycolysis for a minute here. So what I'm going to do is just going to draw you kind of a very brief outline of glycolysis. So remember, glycolysis starts off with glucose, okay, and then there's 2 steps, and we end up with fructose 6-phosphate. So fructose 6-phosphate. And that's where the enzyme is that we're interested in. I'll write that one out in a second, and then you get fructose 1,6 diphosphate, so di for 2. Then we get a few more steps. Not sure exactly how many steps. We'll just use 3 arrows. And then we get that PEP. You may remember that phosphoenol pyruvate was the second last step of glycolysis, and then we get pyruvate. Okay, so there's a very, very simple way to represent glycolysis. You can see we're missing a lot of details, but the important thing is right here is this enzyme here. So the enzyme is phosphofructokinase. There we go. And you can abbreviate it as PFK. So that's the French abbreviation for Kentucky Fried Chicken, right? Poulet. I don't know what the F is for. Are they using fry in French? I don't know. I just know I've seen that with KFC. Okay, so what is the point I'm trying to make here? So it turns out that this particular enzyme is a major regulation site for glycolysis and all of respiration. And so maybe I will say that. This is, so PFK is the major regulatory enzyme of respiration. So you're probably wondering, okay, how is this thing regulated then? Okay, well, it turns out that there's some feedback inhibition on this thing. And it's from that PEP. So I'm going to draw just sort of a line here. There we go. And so that's supposed to be a dotted line and an arrow is what that is. And so this, I'll put a negative sign beside this. And I'm going to draw another arrow. something else that inhibits this thing is actually ATP. So let's write a note about that. Let's see here. I want to put it, I'll write the note here. So if PEP, so the concentration of phosphoenol pyruvate or ATP, what if those things are high? So meaning there's a high concentration of those things. So what if? So what does that mean? It means we have enough ATP. We have plenty of ATP. What else does it mean? It means the cell needs to turn off glycolysis. So we can write that as well. Cell needs to turn off glycolysis. What else is it? So in this case, what happens? Then it downregulates or turns off our enzyme, PFK. So those are signals for that enzyme to be turned off. And if you go back to our notes on enzymes, when we were talking about before the break, I had mentioned we would come back and talk about allosteric regulation. So this is some allosteric regulation of phosphofructokinase. But this is not the only part of this. There's another type of regulation is we can turn this thing on as well. So I guess I meant to put another negative right here just for that ATP. But I want to show you how we can turn this on as well. And so I'm just going to shrink this whole thing so I have lots of room to write. And what I'm going to do is put ADP over here. I'll use a different color. And I'll use a plus. Plus meaning we're turning the whole thing on. So this is me. So let me just put a legend here. So negative. This means inhibition. Positive means activation. Okay, so our note in green, I'm going to just write it up over here. So if ADP, so what is ADP? Remember ADP? It's adenosine diphosphate. So if those levels are high, what is going on? All right, so I'm just going to make sure I spell everything nice here today. So if those levels are high, so what does this mean? So if you have lots of ADP, it means that your ATP is low because you've used it all up. So your ATP concentration is low. What else does it mean? It means the cell needs more glycolysis then. Cell needs more glycolysis. Therefore, what's the cell going to do? It's going to turn on or activate, you could say, our enzyme, PFK. So I'm sure lots of you are looking at this thinking, wow, this is a lot, right? It looks complex. I see somebody has mentioned that's poulet-free Kentucky, only in Quebec. Yeah, I don't know if that's, I guess it's not the same anywhere else. But anyway, yeah, that's what I always think of when I see PFK. So you're looking at this, and we've got this pathway. And what I'm trying to show you is that this phosphofructokinase is a key regulatory enzyme. And it's kind of like the switch. It turns on glycolysis or it turns off glycolysis. It turns on glycolysis when we need ATP. It turns off glycolysis when we don't need, when we have lots of it. And this is very important. It's done way at the beginning of glycolysis. Notice it's not done at the first step. You're probably wondering, why didn't we just do this at the first step? Well, we don't just eat glucose. We eat a lot of different food sources. And so it turns out that this third step is actually a better place to regulate the entire process. So that's what I want you to get out of this. Phosphofructokinase, key regulatory enzyme. We turn it on when we need ATP. We turn it off when we don't need ATP. That's what I want you to take from this. So let's go back to our PowerPoint and continue talking about the steps of respiration. Where is the PowerPoint? There it is. So there's glycolysis. So step number 2 is something called pyruvate oxidation. And actually, maybe what I'll do before I show you this, so I'll go back to our whiteboard, and I'm going to go back to what we were drawing here. So let's take a look at the next step here. And so what I'm going to do before I draw the next step is I'm going to draw a little dotted line like this. You're probably wondering, what is that dotted line? Well, it turns out that this here is the cytoplasm, and everything else happens in the mitochondria. So there we go. So basically, this next step here, our pyruvate actually gets transported into the mitochondria. I'll put a 2 in front of that. I'll balance the equations just because I do want to kind of count up all the ATP we made in the end. So this step here, what do we do? I'm just going to shrink this a tiny bit more. And we're going to follow the carbon for starters, okay? So what we're going to do is we're going to take the pyruvate and we're going to make something called acetyl-CoA. So there we go. We're going to make 2 acetyl-CoA. And I'll talk about that in a minute. Oh, come on. Wants to turn all my C's into O's. There we go. Acetyl-CoA. Now, how do we make acetyl-CoA? Well, it turns out we need something called coenzyme A. So coenzyme A we can represent as CoAsh, and I'll talk about that shortly. And it turns out we also release some carbon dioxide. So there's our carbon. And we're not going to make any ATP at this step, but we are going to make some NADH. So I'm going to throw that in there. We're going to make 2 NADHs. There we go. So that stage there is called pyruvate oxidation. So I will write that down. This is pyruvate oxidation. Right there. Okay, so let's just take a look at what the textbook has, and I'll show you this pathway in a little bit of detail. So you can see this. What they're trying to show is it's getting transported into the mitochondria, and all of this happens inside the mitochondria. This pyruvate oxidation. So notice we have our carbon dioxide is being released. So this little functional group here is called a carboxylate, and it gets released as carbon dioxide. And so that, of course, in our body is going to get expelled at the lungs, right? I'll just say expelled. We have this coenzyme A is coming in, and I'll talk about that in a second. I have it actually a diagram of it on the next slide, and we're making acetyl-CoA. Also part of this process is we're going to make some NADH as well. All of this is multiplied by 2. If you want to keep a count of how many are made per glucose molecule, we want to multiply everything by 2. So this actually all happens in one very complex large enzyme, and it's actually several chemical reactions that are happening all kind of at once. Actually, very fascinating enzyme. So let's talk about this coenzyme A for a second, okay? What is it? That's it right there. So notice this whole thing. Again, look at this. We have basically adenosine right there. And actually, I might have a label on that. I do have a label. That's adenosine. And notice here over at the other end, oops, we've got a bunch of functional groups here. We've got a couple of phosphates. So if you circle all that there together, that would be ADP. It's got an extra phosphate down here. It's got a bunch of other elements over there. And the last thing I want to point out is this thing here, this SH. So notice that this is coenzyme A, and then SH is on the end. So SH is sulfur and hydrogen. So we often abbreviate this thing as CoAsh. And the SH actually do represent a sulfur and a hydrogen out of this whole thing. And that's kind of the important part. So take a look at what happens to coenzyme A when it gets converted to acetyl-CoA. So we end up with this acetyl group here. So an acetyl group is basically this here, CH3, C double bonded O. It looks like that, and it's attached to something. And if you've heard of acetic acid, it has an acetyl group on it. And so what are we doing? We have this giant massive thing, this coenzyme thing, and we're just taking a few atoms, that's exactly what we're doing. Because what else are you going to do with this? This little thing here, this little group, it's not water soluble, and it needs something to basically carry it to the next reaction. And that's all it's doing. So you can think of coenzyme A as a carbon carrier. It's taking those atoms and it's just bringing them to the next step. And coenzyme A is actually involved in a huge part of all sorts of metabolic reactions, doing basically the same thing. It's part of fat metabolism, breaking down fats, part of breaking down proteins and so on. And all it's doing is carrying those two little groups. We're not really going to talk about it too much more, other than it's basically gets recycled in the next step. And that's kind of all that we need to know. So what is the next step? Let me just do a time check here. We're doing just fine for time. I want to talk about the citric acid cycle today, and then that will be where we're going to finish. So the next step is the citric acid cycle. So we did glycolysis, we did pyruvate oxidation. So let's talk about the citric acid cycle. I'm going to flip back to the whiteboard for a second, and I'm going to draw that, at least part of it. So I'm going to shrink everything just a little bit more. Okay. So we'll go to our next step here, and my next step is the citric acid cycle. So what I'm going to do is just draw that like a circle like that. And that's my arrow head right there. So citric, oh, come on. I don't know why it likes to turn my things into circles. Citric acid cycle. Okay, and for the most part, what we're looking at is expelling our carbon as CO2. So I'm going to come back to that in a minute and fill in the other details in a couple of minutes. But just following the carbon, we're going from the acetyl-CoA, and we're making CO2, and the coenzyme A itself is also recycled. So CoAsh right there. Okay, so we'll come back to that, and once we have a chance to look at it here a little bit more. So by the way, you may have heard this called the Krebs cycle. And that's okay. That's just more of an old school name. But the name has kind of stuck. What we're doing is in biology trying to name things a little bit more functionally in terms of what is going on rather than naming them after people. But either are acceptable, of course. So let's take a look at what's going on with the citric acid cycle. So this is, of course, happening in the mitochondria. And I'll come back and talk about the mitochondria a little bit more next day. But where is this happening? This is happening right here in the matrix. And the matrix is this kind of innermost chamber of the mitochondria inside both of the sets of membranes. So up top, you can see pyruvate oxidation right here. We've got acetyl-CoA. It's going in to the citric acid cycle, and we're releasing coenzyme A. And this whole thing, you're going to, this is the quick notes version of it. I'll show you the details in a moment. We're going to release some carbon dioxide. We're going to make some NADH. We're going to make some ATP. We're going to make some FADH2. So we'll come back to that in a second. So let's take a look at the details of this. I guess that slide is a repeat. Okay, so there's our first step here. Our first step is we take this compound here. It's called oxaloacetate. We take the two carbons from acetyl-CoA and we make something called citrate. So citrate, by the way, is the same compound that you'd have in sour candy. So it tastes delicious if you like sour candy. So at this point, not much is going on other than we've taken the carbons and we've integrated them into another compound. And we're going to start breaking things down. Notice we have another mutation going on here where we're changing and rearranging things. So it's called actually an isomerization going on, and we're making something called isocitrate. Really not that important. What's more important is the products that we're making. So notice we have another oxidation step right here. We're making some NADH. There goes the carbon dioxide. We're going to do that again. Carbon dioxide released, making another NADH. So this is good. We're making lots of NADH, but what's so important about that? We'll get to that shortly. Why it's important that we make all this NADH. There's another step down here where we're actually making GTP. You may notice there. And GTP can be converted easily into ATP. It doesn't really matter which you make, GTP or ATP. You could make UTP, you can make CCP. It doesn't matter which nucleotide you use. ATP is just the most common one. Sometimes enzymes use GTP a little bit, and they're kind of considered equivalent. So you can think of this as ATP is being made right there. We're going to make one more electron carrier, our FADH2 there. And then we're going to make one more NADH way over here at the end. So not too bad. We're making ATP, not much of it. And we're making some of these NADHs and these FADH2s. So let me just go back to our whiteboard here for a second, and I'm going to draw some of those things, okay? So remember, we care about our ATP, so I'm going to draw that in there. And so technically, overall, we're going to make, I'll draw it right here. We're going to make 2 ATP per glucose. So 2 ADP plus inorganic phosphate. We're making 2 ATP. So that's kind of the net. And this is what we care about really is the ATP. We're also going to make some electron carriers. And overall, we are going to make, if you go back and take a look at that PowerPoint, we are actually making, 3 NADHs, okay? But we're going to multiply that by 2 because, of course, we have 2 glucose. So we'll throw that in. We've got 6 NAD pluses. That makes 6 NADHs. And lastly, we've got that FAD thing. So it's an electron carrier, remember. So we're making 2 FAD, and we're making them into 2 FADH2s. Okay, so I'll come back to this in a second. If in case you miss anything, we'll come back to that in a second. Okay, we'll go back to the PowerPoint. That's why it's nice to have the PowerPoint and the whiteboard all at the same time. So that's the citric acid cycle. You notice that it starts off with this oxaloacetate, goes through the cycle, and then at the end it regenerates that oxaloacetate. And that's why it's called a cycle, because everything is regenerated at the end, and it's ready to take another acetyl-CoA and do this whole thing again. So overall, we're not making a lot of ATP yet. And that's kind of, one of the things to talk about here in a minute. There's the end products overall. I mentioned there were 6 of these, 2 of these. Carbon dioxide, I didn't really count them, but there'd be 4 carbon dioxide and 2 ATPs. So overall, that's what we're making from this whole thing. And there's the quick and dirty version of the whole thing. And there's some accounting right there. And so it says here, not much energy, or is there? Okay, so what is going on here? And let's just go back and take a look at this thing here. And like I said, I'm going to go back to our diagram here in a moment. So overall, remember what we made. In glycolysis, we made 2 ATP. We didn't make any ATP in pyruvate oxidation. So we could say 0 ATP over here. And we made another 2 ATP in the citric acid cycle. So we've made 4 ATP. That's not very impressive, is it? But remember, we have all these NADHs and NADHs, FADH2s. We have another NADH here. These are all going to go to the electron transport chain. And there we're going to make a whole bunch of ATP. And that's what we're going to get into next day. So let me just go back to our diagram. And for completion, I'm going to add that last thing. So NADH, NADH, FADH, and NADH, these go to the electron transport chain. Transport chain. And if you want, you can write down, they're going to go there and they're going to make a whole bunch more ATP. But that's what we're going to talk about next day. Okay, so I'm going to come back and talk about this in more detail next day. I'm probably going to review all of this, so I'll maybe have to probably draw it all on the board all over again, just to give you an idea of what we're looking at. But it is important that you do follow these things, right? Carbon, ATP, and electron carriers, because those are the kind of things I'm going to ask you about on the exam, on the next midterm. What is going on with glycolysis? Well, glycolysis is making 2 ATP, 2 pyruvate, and 2 NADH. What's going on with pyruvate oxidation and so on. So we'll talk a little bit more about that next day. We'll talk a little bit more about the next midterm as we get a little bit closer to it. Other than that, I guess I'll see everybody in person on Wednesday, and have a great Tuesday.